Hello everyone, and a very warm welcome to today's Partek training session on spatial transcriptomics data analysis. My name is Simit Patel. I'll be your webinar moderator for today. And with that, I'd like to introduce our presenter today, Dr. Ivan Lukic, a senior field application scientist at Partek. Ivan obtained his MD and PhD in immunology from Zagreb University School of Medicine in Croatia. I moved on to do postdoctoral training at the University of Heidelberg in Germany. As a research scientist, he authored and co-authored several dozen manuscripts on multiple roles that the immune system plays in health and disease. Ivan then joined Partek in 2011 and has since advised hundreds of Partek users across the globe on various aspects of experimental design and data analysis. And with that, I'll hand you over to Ivan. Thank you very much.
Thank you so much, Simit, for this super kind introduction. So hello, everyone. Good morning, good afternoon, good evening, depending on your time zone. This is now Ivan speaking, and I wish you a very warm welcome to this training on spatial transcriptomics analysis. I will start by a brief introduction to Partek. After that, I will give an overview of Partek Flow, our analysis solution for multiomics, and then switch to the main part of this session, which is actually data analysis. So Partek is a software company with more than 25 years of experience in data analysis. Our key product is Partek Flow, a comprehensive and powerful genomic analysis platform. When creating Partek Flow, we had different types of users in mind: biologists, bioinformaticians, and large organizations, and we included a wide range of functionalities to meet their respective requirements. With Partek Flow, there is no need for command line or knowledge of a programming language. All the analyses are performed using point-and-click interface. In addition to our own algorithms, Partek Flow ships with a number of commonly used tools, and you can also add your own tools or scripts. Finally, Partek Flow includes user and data management as well as resource management tools. You can install Partek Flow on any hardware, cloud, compute cluster, or your local computer. And then all you need to work with Partek Flow is a browser-enabled device. Here's just a small example of our charts. All the plots can be extensively modified and then downloaded to your local computer as an image file. In terms of assays, we support a wide range of omics applications, genomics, and proteomics alike. You can start your analysis at any point in the pipeline. For example, if you have BCL files or FASTQ files, you can start with those and go over all the processing steps in Partek Flow. Or if your data have already been aligned, you can start with BAM files. We also support quantification matrix files and Sera objects. Furthermore, any result can be exported from Partek Flow in one of the common formats. Say, if you have normalized counts as a text file, no problem, you can get one just with a one-click download to your local computer. A functionality that we recently added to Partek Flow is the search portal. It allows you to perform searches of either your own project database or of the datasets from public repositories, which we are adding to Partek Flow. With this, we've come to the conclusion of the brief overview, and I will now move forward and introduce the training dataset. The data were shared by 10x Genomics to FFP samples. One sample is a normal human prostate, while the other one is adenocarcinoma of the human prostate. Initial data processing was done using the Space Ranger pipeline of 10x Genomics, output of which was then imported directly into Partek Flow. My biological question was to find gene pathways that are affected by cancer. To that end, I performed pathway analysis by comparing gene events between the cancer and the normal tissue. Here's the list of the top 10 upregulated pathways, and they are ranked by the fold change. So now it's time to go live, and I will show you how to reach this stage in a step-by-step fashion. Okay, so this is Partek Flow. It's a browser-based application. We are looking at my home screen. You see the overview of my recent projects as well as what I've been doing over the past few months. Step number one is data import, and I will start by importing gene expression parts of the spatial transcriptomics data. My suggestion is to always start by moving all the files that you need to your instance of Partek Flow using the transfer files tool, this one over here. We have a how-to video on the topic, and you will find it under the help. Under the help menu over here, you will also find link out to our documentation as well as the form to contact us directly. I have already moved all the data, therefore, my only step that I need to do right now is to create a new project, and I will call it Spatial Training. It's already in the autofill list, an empty project, and we are going to get the data. For the Space Ranger output, I need to use this option, import single cell data. Okay, where are those data files? I have already moved them to the Partek Flow server. Therefore, I'm going with the first option. When you click on the browse icon, the file browser comes up. You can either browse through the directory structure, or if you know the path to the directory with your data, feel free to just paste it in here, like I just did. And Partek Flow shows you the supported file types. There are 4 files in this directory, 2 samples, but 4 files, because for each sample, we have the image data and we have the expression data. Our recommendation is to use the filtered feature barcode matrix files. So we'll just select those 2 for our 2 samples and then continue. Next, to start importing the files. Okay, sample table, 1 sample per row. Default sample names match the file names. Let me change them to a more straightforward format. The first file is just normal prostate sample. The second file is the cancer prostate sample. Annotation file, yes, I suggest to add them at the beginning. We need to match the assembly and the gene annotation file that were used upstream in the Space Ranger. For alignment, I'm 100% positive that the data were aligned to AG38, the current version of the human genome. About the gene feature annotation, I'm not sure. We suggest to use Ensemble, and if you don't know the exact build that has been applied in Space Ranger, I would simply use the most current one. A few builds here or there won't make a profound impact on the results. These are the raw counts that we are importing, and I would like to import all the features, all the genes, irrespective of whether they were detected or not. Just then hit the finish button. I won't actually do that because import takes a few moments, well, actually a few minutes, and therefore I will just revert to the old cooking show trick. I have a project where this has already been imported. Let me switch to that, please. And this is exactly what you see after data import. Our first data node, single cell counts, and that data node holds the expression information. Next step is import of the image data. Okay, to work with Partek Flow, you just click on the data node that you would like to work with. Context sensitive toolbox appears on the right. And under pre-analysis tools, let's expand this. I want to use this annotate Visium image. You've seen the data table before. The idea here is to point to the image files, one sample at a time. First one is the cancer sample. It's the same file browser as before. Paste in the path. And this one is the matching image file. Continue. Same thing for the remaining sample. It's the normal prostate image. Continue and finish. Okay. And here we go. This is our result, the annotated counts data node. When you mouse over, the balloon shows you some basic descriptive information. We have 2 samples inside. The total number of tissue spots is just under 7,000, and the total number of features is about 18,000. Now, double click to open the result. If you're not familiar with Partek Flow, this is Data Viewer, our visualization and analysis tool. The canvas with the plots is in the middle, and then the controls are on the right, are on the left, I'm sorry, and on the right. Let's create some more space. I don't need these controls right now, and we will focus on the current sample. We are looking at one of them. The blue dots are the tissue spots, and by changing the opacity of those, we will now use one of the controls on the left, this one. By changing the opacity, we actually switch from expression to histology and back. Just a little bit further down, you will find another control. We refer to these sections as cards that enables you to switch from one sample. Currently, we are looking at the cancer one to the other. So this is the normal sample. And now let's have them side by side. In the upper right, you will find some additional tools. The first one here is duplicate plot. Okay, now I have 2 normal samples. All I need to do is change the one on the right. We'll just switch to cancer. And then to avoid any possible confusion, add the titles. The current plot is my cancer sample. Maybe just increase this a little bit. Okay. And then the plot on the left is my normal sample. Right. Now, I will need this display later. I don't need to recreate it again by clicking. I will just save the session. Give the name of this current data viewer session, something like tissue slices, and save it. Okay, good. Time to go back to the pipeline. And we are ready for the next stage. So now we have the data in, we have both parts of the data, and I would like to process it to keep only the high quality tissue spots for my downstream steps. We are going to apply 2 filters. Here's the expression data. I will start with a single cell QC. Well, these data are technically not single cell, but all the tools can be applied to them. Single cell QC task has no setup dialog. You just launch it. The result can be opened in the data viewer. Double click. And we see 3 plots over there showing number of counts per tissue spot, number of detected features, detected genes per tissue spot, and fraction of ribosomal counts per tissue spot. Within these plots, each individual dot is a tissue spot, and the distribution of the metric shown on the y-axis is shown by the violin, the gray violin in the background. Now, I said that I wanted to keep only the high quality tissue spots. Therefore, I'm going to remove only the outlier ones using the selection card on the right. When you mouse over one of these blue handles, Partek Flow will show you a histogram with the data distribution. This you can use to help you guide your decision, or you can simply use the to actually select. You can either move the handles. As you see, the effect is immediately visible on the tissue spots, or you can type in your cutoff values. You can combine the criteria. Let's do something like this. And perhaps one final touch on the ribosomal counts. Just trim a little bit. We are ready to go. When I click on the funnel icon, Partek Flow will recalculate the plots, show only the selected tissue spots, and the final step is here, apply observation filter. So do the filtering. The next dialog is select data to filter, and I want to select my single cell counts data. Okay, the task is in the queue. Let's go back to the pipeline. We don't need this data viewer session for later. Okay, this task is now in progress. Mousing over gives you the overview of the current state. We don't have to wait. I can immediately set up my next task. It's going to be gene level filtering over here in the toolbox, filtering, filter features. There are 4 feature filters to choose from. Please note that this word over here, documentation, is actually linked to our documentation page, which provides more detail on each of those filters. And you will also get some information by mousing over the tooltips. For this analysis, I want to apply a noise reduction filter, basically exclude the features which were not detected or where gene expression is just at the background level. Multiple criteria are available. I will switch to the value. So exclude the features where value is equal to or lower than 1 in at least, let's go with 99% of the cells. And finish. Okay, the previous task, tissue spot-based filtering, has completed in the meanwhile, and we'll take a look at the result by double-clicking on the task node. There are 2 tables available for you. The top one summarizes the expression values. The lower one shows us our filter criteria. Remember, we did some filtering with respect to the number of genes, number of reads. So here are the filter criteria per sample. And finally, the summary statement, we kept in just over 99% of the tissue spots. And it was a very gentle filtering. We're going back to the pipeline. Let's take a look at the result of the gene filtering. Again, table with some key expression metrics, and then the box plot showing the distribution per sample. I won't go into detail right now. We'll come back to the box plot. Just over here, the summary statement, we kept just over 50% of the genes. And going back to the pipeline, this is now my clean data set over here. Next step is exploratory analysis. I would like to get a feel for my data. I would like to take a look at the structure, maybe detect any trends. Please remember that these are still raw counts. Therefore, we are going to normalize the expression values. We have a number of normalization transformations available. In the first pass, I will use the tool at the top, which is the most flexible one. Yeah, about flexibility, we offer a number of transformations. And if you're not sure, if you're just making your way through your first spatial transcriptomics dataset, why not use the recommended one, which is very gentle, works really well for instance, single cell data. It's CPM with some log transformation on top of it. And finish. Once I have the normalized values, I would like to visualize the data. For that, I need two steps. First, we are going to run the PCA to reduce the number of dimensions. PCA, Principal Components Analysis, is here under Exploratory Analysis. There is a setting I would like us to take a look at. This one, Split by Sample. If the checkbox is on, then PCA will run on one sample at a time. When you turn it off, the data are combined, and then you run PCA on it. For your actual projects, you probably want to do both. Right here, to save us some time, because we have barely the time to scratch the surface, I'm going to combine the data in one large set and run PCA on that. So that's finish. And for visualization, I will not use PCA, but I'm going to turn to UMAP, and you can find it under Exploratory Analysis. In this training, or for this particular data set, I decided to go with default settings. Finish. We are not going to wait for these two tasks. Instead, we will just do the cooking show trick. And here we go. Our UMAP is ready for inspection. Double click to open it. Each dot is a tissue spot. Our UMAPs by default are showing 3D with a lot of flexibility, a lot of interactivity. We see two clusters over here. And I guess that the data cluster by sample. To double check for that, we will start working with the data card here on the left-hand side. The data card shows all the steps of the analysis. These are our results, our data nodes. If you mouse over a data node, you will see a thumbnail with a preview of the pipeline. Alternatively, if you click on a data node to open it, a card will roll out with the available attributes as well as with the available features. Speaking of attributes, I would like to color my samples by sample name. It's just simple drag and drop. Yeah, and there we go. Tissue spots from the normal sample cluster together. They are in red, and so do the tissue spots from the cancer sample. Now with cell culture, with cell lines, this would have been fine, but over here, I expect that the cancer sample will contain some normal tissue, which is inevitable while harvesting these samples. And that normal tissue from the cancer sample should cluster with the normal tissue from the control sample. We don't see that here, but what we do see is batch effect. Now, I would like us to do something about it. We will go back to the pipeline. I won't jump straight into batch correction, but first we are going to consider one step upstream. That's data normalization. So let's just for a second consider normalization. As you know, data normalization is really critical because it has profound impact on any downstream steps. So why don't we compare the procedure that the CPM transformation, the procedure that I used already, with an alternative normalization. Here under normalization and scaling, I will use SC transform. It's a part of the Sera package. And if you take a look at the balloon, it tells you briefly what the method is about, that it used a negative binomial regression model. Run SC transform with the default settings and finish. Let's just fast forward, shall we? That's the same project, but the SC transform has already completed. And there's the current active data node, the SC scaled data. Okay, effect of the normalization. Let's double click this node. We've seen this before, but now after a normalization, there are two plots side by side, box and whiskers showing distribution of expression values before and after normalization. It looks, at a first glance, it looks fine after normalization. However, when we mouse over, the balloon will give us key percentiles for each sample, one and the other. And looking at the plot, we don't really see the lower whisker, right? We don't see the full box, and yet that can be easily explained by looking at a percentile. So here, most of the data, or half of the data, are down there with the median and the lower quartile and the lower whisker. In the other words, data are kind of skewed, and I don't really like that. So let's take a look at the result of the SC transform instead. That's over here. Box plot after normalization. There is some difference between the cancer and the normal sample in terms of the distribution, but the upper quartile and the lower quartile are quite comparable. And besides, now we see the box and whiskers. It's even distribution of the data values. Please note that with SC transform, you can end up with some negative values, which is expected. For me, my decision was to actually use SC transform in this particular project. Just to quickly summarize this section, I advise you to take a look at the normalization carefully and to make an informed decision how to proceed. It's not easy to give an upfront advice. It actually depends on your biological question. It depends on your results. So you need to visualize the data, and here's how you can do it. With that, we are ready for the next step, which is a removal of the batch effect. We have 3 tools available. You've noticed the batch removal section in the toolbox. You will find 2 tools there: general linear model, which is ANOVA in reverse, and then Seraph reintegration, another member of the Seraph package. And then for the third tool, it was designed to run on PCA result. Therefore, you need to select the PCA data node, and then you will find Harmony tool over there. Since I decided to use the SC scaling in the current project, I then simply used the SC Seraph reintegration because it's a matching batch removal. Seraph reintegration is a batch removal designed to work on SC transform normalized data. What Seraph reintegration does is identifies data points in same biological state, such as tissue spots across my samples. Those are anchors, and then those anchors are used to correct for technical differences. On the setup dialog, we can pick the factor which we want to remove from our data. I would like to remove the sample factor. And once I get my corrected expression values, batch removed expression values, I will find them here in the integrated counts node. I want to do the same thing as up there. I will visualize the data by running PCA with the default settings and then using UMAP to actually plot. So here's the UMAP. Again, right here, I was happy with the default settings. Yep, let's save some time by using fast forward. And let's take a look at the UMAP. It looks interesting. There's only one cluster right now. It's in the form of a hammer. Let's see about the outcome of the batch removal. Remember before, 2 samples clustered separately. Do the same thing. Open the starting data point where my sample names are and just color the data by the sample name. Okay, what we see right now is that there are some tissue spots from the cancer sample that cluster together with a normal sample. So what I guess is going on here, and I'm going to double check it later, is that over here, the normal tissue from the cancer sample clusters together with the normal tissue from the control sample. And that's what I wanted to see. And therefore, having my batch effect corrected, I will now move forward to the next step. I would like to compare normal tissue relative to the cancer tissue. In other words, we're now going to talk about classification, right? So how can I classify my tissue spots into cancer or normal? There are several ways to do that. For example, I can be guided by my exploration. Say, okay, over here, these are my cancer tissue spots, and then I'm going to revert the selection, and these are my, this is my normal tissue. Or I can use an algorithmic approach to first detect any structure in the data and to make the decision, okay, where to draw the line. Let's do that. Going back to the pipeline. So these are my data after batch removal. And I'm going to use a clustering method over here under exploratory analysis. Under exploratory analysis, you are going to find many clustering tools such as graph-based clustering, k-means clustering, or hierarchical clustering. The one that I would like to use is graph-based clustering, very common with single-cell data. And please note that we suggest that you run it on the PCA result. Okay, so here we go. Under exploratory analysis, let's do some graph-based clustering. At the same time, I would like us to get some biomarkers. So when I get my clusters, I would like to see what is specific about them. What makes cluster 1 different from cluster 2? And biomarkers, so specific genes will answer that question. So finish. This doesn't take long, therefore I'm actually going to wait for it. Here we go. Now, back to the UMAP. I want to see some color, my graph-based clustering result. Over here on the data card, we can either look for that result by simply browsing through, or why don't I use the search box, because I know that I'm looking for the graph-based. So here it is, graph-based clusters. Open the card, and my graph-based clusters are over here. To color, again, it's drag and drop. Okay, and I talked about, so how many? 15 clusters, okay? And then I talked about the biomarkers, meaning I need to add a new element on the canvas. That's a table with biomarkers. First, add a table, and then specify the data for the new table. Here, biomarkers. Data viewer shows you the top 25 biomarkers, but you can get the full list by clicking on this icon in the top right, export table data. Now, the genes in the table are linked back to the UMAP. Here's in cluster 2, one of the top markers is SPON2. That's very interesting. That's one of the markers of the prostate cancer. So let's color the UMAP by SPON2. Here's the legend. High expression level of SPON2 is detected in this handle of the hammer where I believe the cancer tissue is. Okay, now one way to quickly check for that is to actually split the UMAP in two. I perform PCA on both samples at the same time, but we can split them right now. Remember my sample name. This time I will not color, I will split by sample name. And okay, all the SPON2 positive tissue spots are in the cancer sample. Right, we can merge this plot, and I will merge these plots. The easiest way to do it is to use the undo button. Okay, let's bring the tissue data into play right now. I'm going to create a new data viewer session. Well, actually, I don't have to do it from scratch. Remember that I saved the data viewer session before. At the very beginning, our tissue slices, let's reopen them. I want to check where the SPON2 expression is located. I don't need to use the biomarkers table. I can access the same information from the data card. After batch transformation, the data node is called integrated cell counts. It's here, integrated counts. Gene data, let's search SPON2. Here it is. Just add to one and add to the other slice. The scales or the range of these scales is not the same. Look at the upper boundary. I need to change this quickly, customize it. So on the normal sample, going back to the color card, I would like to set the, right this. On the normal sample, I would like to set the max to 13.7. Now they're on the same scale and we can clearly see a hotspot of SPON2 right in the middle of the cancer tissue. And we can take this one step further, add the UMAP information. Adding a new UMAP is easy. It's drag and drop right here on the right. Where is the data? That's UMAP. Okay, but I have two UMAPs, no problem. We will just inspect the pipeline. So not this one, but the one after batch correction, right? There is something with the, I wanted to use clustering to actually pick my normal tissue and my cancer tissue. So going back to the data card, where's the graph-based clustering, graph-based clusters. Now, you know that I can color UMAP by cluster, and we don't need the legend, but we can do something else. We can use the graph-based clustering result and land on the selection card to select the data points by clustering result. Okay, let me show you what I mean by that. I'm going to uncheck some of the clusters, and when I do so, we'll see they're unchecked on the plot. So all the plots are connected, say 2 and then 4 and then 10, right? Those are tissue spots in the handle where we saw the high expression of SPON2, and also note take a look at the legends. The tissue spots with a high expression level of SPN2 were removed. Okay, meaning that the remaining or the currently selected tissue spots represent my normal tissue. And I want to classify the current selection as normal tissue. Okay, and then the rest, invert selection, is just cancer tissue. Okay, now I'm going to apply these classifications to my data. Select input data, that's the integrated counts. Select attribute name, I will call it tissue type. I don't need biomarkers right now. We can always launch the task later. Run. Okay. Going to the PCA, going to the pipeline. Here. That's my result of my classification. I basically added a new attribute, cancer tissue, normal tissue, and I can use it for the next step. The next step is differential expression. What I would like to use now, or to do now, is to compare my two groups, cancer tissue and a normal tissue, run some statistics, and find differentially expressed genes. Starting on the integrated counts, there are many statistical tools under differential analysis. The one that I will use right now is GSA. Basically, under default settings, it runs limit trend. Partek flow is very flexible. You can use any number of categorical or numeric attributes. Right now, I'm working with my tissue type. Next. And my question, my contrast of interest is cancer versus normal. Add comparison and finish to run it. We are running this live. And the result is a gene table with genes on rows, and on columns, we get the p-value and the fold change. Now, these two pieces of information can be visualized using a volcano plot. Here's the icon display volcano plot. Upregulated genes in red, downregulated in blue. Like all our charts, the volcano is also very interactive. For example, you can annotate the genes directly on the volcano. There's another plot that's a violin plot. We will now focus on the filter panel here on the left. For example, let's look for one of the marker genes for a prostate cancer. And the gene is KLK2. There it is, highly significant. And a path to the violin plot goes through the dot plot icon over here. When the data viewer comes up, it will show the dot plot, but customization is very easy. Why don't we create some more space here on the left-hand side by hiding the bits that the cards that I don't need. On the summary card, just hit the switch, add the violins, and we can do more, such as overlay the dots on the violins, add some color. And there's something else that I want to do. Notice the order: first the cancer sample, then the normal sample. I want this other way around, just drag and drop. Normal first, then cancer. I'm happy with this image. And then to download it to my local computer, there is the save image icon again in the upper right. Let's do some other filtering. Clear the filter. Now I would like to use the to come up with a list of significant genes by using the usual criteria such as FDR. Let's do 0.05, about 8,000. Add the fold change. This is a cancer experiment. So let's do ±4. Okay, just under 800, and then generate the result. Okay, show me the significant genes. Now we have our list of significant genes, and it's time to go to the next step of the analysis or next stage. That's biological interpretation. Now I'd like to learn about these genes. First, I'm going to perform pathway enrichment and to look for overrepresented pathways over here. The database that we use for pathways is CAG. Just click on finish. Here's the result. Pathways are ranked by the enrichment score. The higher, the better. And one way to interpret it is to actually take a look at the rankings of the pathways. Going from the top, what do we see? Like focal adhesion, okay. Then extracellular matrix receptor. And then a little bit further down, more adhesion. My take on this is that most significant pathways actually include the genes that are involved in interaction between the cells and their surrounding. And yes, this fits the biology of the prostate cancer. Cell adhesion molecules are very important for the development and progression of this type of cancer. In addition to the table, by clicking on the pathway ID, you get a nice pathway image, upregulated genes in red, downregulated in green, and you can get that image to your local computer, of course. So pathway enrichment is one possible way of biological interpretation. There is an alternative route. I remember that when we perform pathway enrichment, we were running it on a filtered gene list, meaning that our pathway enrichment result is affected by the decision on how to filter. There is another approach. We can perform something that we call pathway ANOVA. You can find it over here under pathway analysis. Pathway ANOVA compares entire pathways across conditions. So unlike the analysis that we just performed, where genes were compared across groups, now we are going to compare entire pathways across groups. Using this same tool, you can also perform gene ontology ANOVA. It's the same thing, it's just the way you organize genes into groups. Pathway interpretation can be more involving. Some of the pathways can have hundreds of genes. Therefore, I'm just in the first pass analysis, I'm going to limit my results to the pathways of this size. Run next. The factor, biological factor, or the experimental factor of my interest is tissue type, of course. And I'm comparing cancer tissue relative to the normal tissue. Add comparison and finish. And this is basically the last step of the analysis that we are going to perform during this training session. Just like the gene table that we've seen a couple of minutes ago, but this time pathways are on rows, and for each pathway we get a p-value and the fold change. Now, some of these we've seen before with the enrichment, but some are new. For instance, TGF-beta. That's important because TGF-beta plays a major role in prostate cancer as it controls apoptosis and cell proliferation. Let's not forget that the chevrons are sort tools. Therefore, I'm going to show you the top upregulated pathways, and you have seen their list before at the very beginning on the slide. Let's take a look at closer look. Let's have a closer look at it right now. So fat, bile acid, linoleic acid, alpha linoleic acid, 7 out of 10 of the top pathways basically revolve around lipid metabolism. This was a surprise for me because I don't have expertise in prostate cancer biology, but it turned out that yes, lipid metabolism is dysregulated in prostate cancer. And furthermore, these pathways are potential therapeutic targets. So a valuable result that we obtained by using an alternative tool. Going back to the pipeline, I told you that pathway analysis was my final step. Therefore, I would just like to summarize. I've shown you how to import spatial transcriptomics data. After that, I performed some filtering, some data processing. The next step was exploratory analysis. I also mentioned normalization and batch correction. After that, we've been doing some clustering, data classification, followed by detection of differentially expressed genes. And finally, I've wrapped up by performing biological interpretation. So start to finish analysis in Partek flow. I hope this was informative. I hope this was interesting for you. And I would just now like to thank you for your attention. Thank you.
Thank you very much, Ivan. That was certainly very interesting and very informative. I certainly learned a lot, so thank you very much for that. And thanks again to our audience for your kind attention. With that, I will draw this session to a close. If you want to reach us, then the best way to contact us is via our website, partek.com. You can fill out the correct form there to contact us. So thank you once again. Have a great day, and we'll speak to you soon. Thank you.
Thank you so much. Speak soon. Bye-bye.